Thank you so very much, and I'd just like to start by thanking Simon and colleagues for the privilege of coming here for your first meeting. It's great to be here. It's great to see old friends like Ahmed, David, Channing, and get the wisdom of Ian, hopefully, a bit later on. So anyway, I'm very pleased to be here. And I just want to start by actually echoing some sentiments that Channing touched on a moment ago. I mean, as I hope I'll be able to persuade you in the next few minutes, the major challenge about high-grade serous ovarian cancer actually is about molecular pathology and the lack of a good classification. You've seen a great example around endometrial cancer, but we fundamentally need the close join between the trials community, molecular pathology, and more molecularly leaning oncologists like myself to make a difference. And it's not going to happen without those synergies coming between the groups, and luckily in the ovary field that has been very productive. So what is high-grade serous ovarian cancer in just a couple of introductory slides? So the first thing to say is that the word ovary is a little bit of a misnomer, because I think almost the vast majority, we don't have evidence against this, but it is likely that the vast majority arise not from the ovary but from the fallopian tube epithelium. This is a disease that is exquisitely defined by p53 mutation. This is going to be a constant theme throughout this talk. And indeed, this is Ahmed's paper when he was in Cambridge, which really showed for the first time that the mutation rate in high-grade serous ovarian cancer approximated 100%. And the consequences of that early loss of p53 are profound chromosomal instability. Now, in terms of the disease, there's a couple of paradoxes. Despite presenting with advanced disease, patients actually do relatively well with standard of care chemotherapy. But unfortunately, survival has not changed over the past 20 years and is dismal. So 5-year survival is around 30%. And what is clear is that the mutator phenotypes, that is, the pathways that are deranged, are likely to lead us to therapies. The best example, I'm not going to talk about it in detail today, is about BRCA1 and BRCA2, germline and somatic mutations and the predictive effect for use of PARP inhibitors. But there will be other mutator phenotypes that we're yet to find. So just very quickly, these are just micrographs through the fallopian tube from this very nice paper by Ronnie Dratkin. But you can see here perhaps that there are ciliated cells in the fallopian tube and of course secretory cells, and the cell of origin is actually the secretory cell. But as you go from normal tube to invasive serous carcinoma, you accumulate p53 protein concomitant with the acquisition of mutations. And perhaps the last thing to say, which is very important, why do most women with high-grade serous ovarian cancer present with advanced disease? Well, you've got the fallopian tube fimbria, these finger-like projections, malignant cells detaching and then just being able to drop straight out of this hollow-ended tube into the abdominal cavity. And perhaps not surprisingly, these high-grade lesions distribute around the abdomen very early. So really trying to pick up early diagnosis in the way that Rebecca presented to you earlier on with a sponge is going to be a huge challenge in high-grade serous ovarian cancer. And then just like esophageal carcinoma or adenocarcinoma of the esophagus, this is a copy number-driven disease. So these large meta-analyses of the TCGA data show this very clearly. This bar plot here is showing the difference between M-class and C-class cases. C is for copy number, M for mutational burden. And ovary here, which is high-grade serous ovarian cancer, is the absolute outlier in terms of copy number, and has only a median number of mutations of a non-synonymous type. And then, rather like other C-class tumours, including small cell lung cancer, some squamous cell carcinomas, esophageal carcinoma, you can see common patterns in terms of loss of HRD pathways, loss of NF1, and MYC amplification. So what's the current state of high-grade serous ovarian cancer at the moment? What actually can we do in the clinic? And the answer to that is the most important thing we do is diagnose patients with germline mutations in BRCA1 and BRCA2, because we know now that those probably account for about 10 to 12% of all women with high-grade serous ovarian cancer. And in Cambridge, Mark Tishkovitz has led a groundbreaking study called HGSOC, where we've changed the way in which that testing proposition is put to women, and in particular when a woman with high-grade serous or endometrioid ovarian cancer see either a gynae oncologist or a medical oncologist or a clinical oncologist, we now have a pathway that allows their testing to be done in a very lightweight and cheaper manner. In other words, all patients are offered treatment. So this plot here is the 100% of all women with high-grade serous ovarian cancer. This arrow is showing decreasing sensitivity. BRCA1 and BRCA2 patients have greater sensitivity and better outcome, although the majority are still not cured. This grey box indicates less confidence in information, but there's a small group in this sort of 1% box of other homologous recombination pathways, particularly RAD51, BCD, BRIP1 and others. Their relationship to outcome is not entirely clear, that's why it's grey. And then at the other end of the scale, as a predictor of resistance to treatment, which is a pretty important thing if we could bring that into the clinic, is cyclin E amplification. And the information here is actually fairly solid, but we do not have copy number estimation as a routine assay in most specialized centers trying to carry out personalized medicine. Well, what about other measures we can do on tumor tissue? So, of course, as you heard earlier on, gene expression has been very important in dividing up breast cancer. It is not, I think, clinically useful at the moment in high-grade serous ovarian cancer. There are 4 reproducible expression signatures shown here: differentiated, immunoreactive, mesenchymal, proliferative. And clearly, the immunoreactive is now of great interest because this correlates with CD8 intraepithelial infiltration. But the problem with these expression signatures is they've been done out with detailed quantitative histopathology and then also non-exclusive. In other words, patients are shown here in columns, classification is shown in rows, so I could be both differentiated and also proliferative here, P, M, I, D, counting for these. So that's a major challenge, and I think the expression profiling is not taking account of the true pathology of these cases, particularly in terms of these other tumour microenvironment components. And then large-scale meta-analysis by Mike Birra and others has actually shown some genes that are prognostic, not predictive, as in BRCA1 and BRCA2, but those also don't have a clinical role. And then larger consortia, such as the OTTR-OCAT consortium, are now looking at these and other markers in large-scale tissue microarrays. And I think ERPR has been reported on, P53 and P10 and CD8 are just coming out now. And these are going to have relevance in terms of trying to find diagnostic subcategories. There's one other thing just to highlight about the challenge of now personalizing treatment with women with high-grade serous ovarian cancer, and that's the rapid move over the past decade to almost exclusive neoadjuvant chemotherapy across most centres, certainly in the UK and across Europe, leaving aside Germany. And that is that most patients present with advanced disease, stage 3 or stage 4, and will have neoadjuvant chemotherapy, and then have interval debulking surgery or delayed primary surgery. These are 2 terms meaning the same thing. That means that as oncologists, what the material that we and the pathologists have to deal with, of course, is relatively limited, because in most cases that is an ultrasound-guided biopsy, a blood test, possibly some ascites if you're forward-looking and you're collecting it, and a germline DNA sample. And the samples that we're getting at interval debulking surgery are, of course, low cellularity tumour, because the patients who respond well will come to interval debulking surgery. Patients who do not respond well will not come to surgery and you're back to a biopsy. So a lot of this means that we have to focus, I think, on liquid and small sample preparation for these assays. So, to set up the rest of the talk, what are the things we're missing for the clinical management? What are the things we've really got to solve in the next few years to make a difference? Well, as I've described, this is a copy number driven disease, so we don't have easy oncogenic measures. We can't simply sequence EGFR or KRAS or BRAF, they aren't relevant. We are lacking a very clear understanding about what the transcriptional drivers are for this disease. We do not have a molecular classification that's worthwhile, and we don't have a very clear understanding about how heterogeneity is making a difference in the disease. And I think key to addressing these problems is P53. So I put it here as the central gene because it's mutated very early on, and we have used this in our work over the past years to look at 3 different properties. One is a tissue biomarker, second is a response marker from circulating tumour DNA assays, and now we're looking at it as a clonal marker, particularly to unpick heterogeneity and also to derive clinically relevant copy number estimations. So I won't spend too long on the sequencing technology, but just to say that we obviously got very interested in this after the work that Ahmed started in the lab, but our big step forward was to develop something called tagged amplicon sequencing. In other words, very low cost, next generation sequencing using a microfluidics platform. The main idea here is you can take very degraded DNA, either from plasma or from FFP material, or high quality DNA, it doesn't really matter, but with a pre-amplification step, and by using singleplex PCR in its original it's in its original development, we could then get barcoded PCR products out that allowed us to sequence in the entire region of P53, entire region of P10, and other hotspots. And we can do that extremely cheaply. So over the last 5 years, what we've used this technique for is really to build an extremely cheap platform for characterizing patients both in the clinic and in clinical trials. This is the basic panel. We've extended this now to detailed germline sequencing for all of the homologous recombination genes. And now we have panels where we can do somatic sequencing and germline sequencing and plasma sequencing. In fact, we're almost getting to a stage now where tissue is not quite so important. I mean, I'd say that with my tongue firmly in the side of my cheek. Tissue will always be important, but we are getting to very good assays that can be done off liquid biopsies. And as we've gone from left to right on this curve, what we've also done is mechanized this and taken it to high scale. So we have now carried out over 5,000 germline sequences and over 1,000 somatic sequences using tagged amplicon sequencing, and are now bringing it into our clinical labs for research and clinical trial studies. And the cost of doing this assay is around £20, comprising 2 technical replicates. So £20 for a somatic sample, £20 for a germline sample, and a little bit more for the plasma sample because of the requirement to extract the DNA is a little bit more expensive. This was touched on earlier on. Analytical reliability around P53 remains poor. And of course, if we want to say something about high-grade serous ovarian cancer, we want to get the diagnosis right. And Challenge showed some of this, but I just want to highlight a couple of things, which is that we have done work looking at different antibodies and different protocols with Martin Coble, pathologist in Calgary, who led this work, really to try and find the most sensitive and most reliable assay for P53 using IHC. This schematic here just shows tissue microarrays stained with different antibodies, and then along the rows here are the different classifications of staining that we have now described. Complete absence, which Channing talked about earlier on, wild type, overexpression, and this under-recognized, certainly under-reported phenotype of cytoplasmic staining, in other words, where P53 is both in the nucleus and in the cytoplasm, and even myself as a pretty awful pathology can see the difference between this and this. But I mean, the take-home message from these very detailed studies where we've combined the best antibody against the sequencing is that if you've got abnormal staining, P53 IHC is a very sensitive and specific marker of mutation. From a clinical perspective, though, if you see what you think is wild-type staining, particularly in a case where you're worried about low-grade serous, which is a different disease to high-grade serous, you really have to sequence. And that's because there are a small proportion of mutations, around 7% of high-grade serous cases, that have loss-of-function mutations, but still produce enough protein to be detected by a sensitive assay. And that's one of the reasons I was asking the Rebecca question in the previous talk, because that means that you're going to score these cases as wild type, but they've actually got a mutation. So you've got to be careful about that. And you've also got to be careful when you're using these biomarkers in clinical trials. So we are participating in one of the first trials in high-grade serous ovarian cancer of a P53 refolding medicine. This is APR246, or PrimaMet, as it used to be called before it was improved for clinical use. We're bringing both circulating tumour DNA and the IHC assays to the trial cohort. This is the first 24 patients here. The study population is those with a non-synonymous mutation in the DNA binding domain, shown in red here. But of course, what happens is when you do the analysis of the phase 1 data, we pick up other cases. This case was probably wild-type expression and was thought to be overexpressing, so not quite sure how that got into the clinical trial. But also we're picking up patients with cytoplasmic staining, shown here and here, which is not being recognized by the wider non-specialist pathologists. Now, refolding medicines might work well in cytoplasmic as well as they do in non-synonymous, but we need the larger cohorts, and we're doing that analysis now. So in the next just chunk of the talk, I just want to talk about what we've been doing with circulating tumour DNA assays. So We've shown previously that we can see evolutionary change in tumours, particularly the acquisition of kinase mutations in EGFR and the acquisition of RB1 mutations. But in the past few years, we've really been focusing on how we could bring circulating tumour DNA into a trial environment. And there are really two key questions there. How does circulating tumour DNA correlate with the volume of disease? And how does it correlate with response? So this visual abstract is just showing work that's about to come out at the moment. We took 40 retrospective patients, 318 blood samples. We had the p53 mutation for all the cases, and in this case we didn't use direct sequencing, tagged amplicon sequencing. We used a very sensitive assay, digital PCR, although the two of those are now very similar in terms of their sensitivity. We compared them against 3D reconstruction of tumour volume from CT images, and we also looked at treatment, and you'll see some graphs that look like this with coloured rectangles indicating chemotherapy course 1, chemotherapy course 2, comparing against CA125, which is the gold standard protein measure for response as a blood marker, against ctDNA. So this is very time-consuming work. Doing the 3D reconstruction was carried out by our radiologists, but the problem with high-grade serous ovarian cancer, it tends to be a surface disease. So in contrast to, say, lung cancer, where you have nice round things you can measure in the lung, you occasionally see round things. These are lymph nodes, but the majority of disease is plaque-like, so you have to reconstruct it and measure the volume. But what was interesting when we did that was we started to get a cut point for the detection of ctDNA from patients with both upfront and relapsed disease. So these lollipops here indicate the tumour volume on a log scale. Open and closed circles indicate the differences between detectable ctDNA in black and open lollipops undetectable. And then there's some dotted lines here, meaning I think this is the wrong way. Yeah, sorry. So dotted lines, newly diagnosed patients, in other words, untreated, whereas the solid line indicate relapsed patients. So by looking at this curve, we're getting down to about detection of circulating tumour DNA when you've got about 35 centimetre cubed of tumour volume. And then if you ask the question about how well that's correlated with the level of ctDNA, that's shown here. So you've got tumour volume along the x-axis and the p53 mean allele fraction shown here, and CA125, the gold standard marker. So the correlation in general is better for ctDNA, slightly worse for CA125, well, significantly worse for CA125. Another important question for trying to use ctDNA as a biomarker is how fast we can measure response. In other words, can we start to do sequential trials where patients are started off on one medicine and then move quickly to others as we try and work out what is working for them. So circulating tumour DNA has some attractive properties in that regard. Firstly, the half-life of normal DNA in blood is around 25 minutes, whereas the physiological half-life of CA125, or rather the clinical half-life of CA125, is several weeks at least. So this plot here just shows the comparison to the nadir value, in other words, the lowest level of measured circulating tumour DNA across all the patients under treatment. So ctDNA again shown in little blue squares, CA125 in yellow-orange circles. And you get a sense that the rate of fall is faster for ctDNA, and that's also shown here in another way, just using box plots, showing that the time to nadir is significantly different for p53 MAF ctDNA as opposed to CA125. Well, what about treatment? Do we see any prognostic or predictive value from treatment? Well, yes, we do. This is, of course, a small sample set, 40 patients, 300 blood samples, so take it with a pinch of salt. But if you look at the ROC curve and ask the question, how much ctDNA predicts time to progression, what we find is that if you fall by 60% after your first cycle of chemotherapy, not surprisingly, you can split the survival curves. So I'm being very guarded here. I think what you'll often hear is people say, oh, well, we've got a test that shows that we can predict some aspect of survival, whether it be time to progression or overall survival. This is by virtue of the data set overfitted, but it does say that there may be a signal, and I think that's important. Very quickly, just one other point which I forgot to mention here is these correlations go down if you have patients with ascites. So ascites is the collection of tumour fluid within the abdomen, and CA125 values in the blood are also affected by ascites. But in fact, if you look at ascites, it also has, not surprisingly, high levels of ctDNA or cell-free tumour DNA, and probably we should view ascites as a third space for circulating tumour DNA. So we are now currently taking these assays to clinical trials in which circulating tumour DNA is the endpoint, and this rather sort of hand-waving question about whether it's truly a response measure will actually be answered in patients that are very homogeneous in terms of their cohort. So just in the last portion of the talk, I want to talk about this problem. So elephants do very well in terms of cancer because they have 20 copies of P53, give or take. But the elephant in the room in terms of high-grade serous ovarian cancer is why aren't we measuring copy number in patients, and why haven't we done it across large patient cohorts to understand what that will tell us about the disease? And many of you may know that the Genomics England effort, which is occurring in England, will bring, we hope, whole genome sequencing to about, well, the numbers change, so I have to be a little bit careful, but originally envisaged as 2,000 patients with ovarian cancer and 2,000 patients with other subtypes. The challenge with this, though, is really getting a clinical report on a whole genome sequence in a meaningful time. And I think Illumina will solve that problem, but at the moment, GEL is struggling with it. Are there other things we can do? And our underlying hypothesis is that the pattern of copy number change probably predicts quite a lot about the disease and its outcome. I showed you a moment ago that this is a copy number disease, so we really should study copy number. If we're not doing it, then we're probably missing something very important. And Rebecca and others showed you this very important paper here from Mike Stratton and colleagues that showed that mutational signatures, particularly the spectrum of mutational change, is associated with different mutator phenotypes, particularly BRCA1 and BRCA2. So we've been worrying for some time that we should be able to develop a molecular classifier for high-grade serous, really based around the hypothesis that the copy number change will drive this. And we were delighted then when Bauke Istra published this important paper in 2014, in which he showed that it was possible to do shallow whole genome sequencing to get copy number change and to use degraded DNA such as that from FFPE. I'm not going to talk about this in detail, but the really neat advance here was combine single-end 50 base pair sequencing with some clever bioinformatics to remove problematic regions. So what we have done is we've taken that approach of doing whole genome sequencing, shallow whole genome sequencing, across clinical cohorts that we've been collecting to ask about what is happening around heterogeneity and evolution over time. So myself and Ian McNeish, my co-investigator in Glasgow, have now collected about 250 patients at relapse with a biopsy and a circulating tumour DNA assay. But we don't just want to do relative copy number. Bauke's original publication shows what relative copy number is. That, in other words, you just count the reads across the genome, you bin the reads in 50 or KB windows, 50 or 100 KB windows, and then you normalize those reads to a median and then say, well, look, there's relative differences. We're interested in something different, which is absolute copy number, which is there 0, 1, or 2 copies of that gene, or are there more copies of that gene? And that's critical because if we want to do accurate copy number comparison as a classifier, we need to have that reliably. If we really want to know what a loss is, we need absolute copy number. And if we want to start pulling out things that might be prognostic, that has to come from absolute data. And our underlying gut feeling is that the methods that are used to do that on, for instance, SNP or ACGH data or whole genome sequencing data probably underrepresent copy number change in diseases like high-grade serous ovarian cancer. And that's because they are, the prior is around a diploid genome. But as has been explained a couple of times in the previous talks, early loss of P53 leads to endoreduplication, and you most certainly do not have a diploid genome in high-grade serous ovarian cancer. And lastly, apart from wanting all that, we want something very cheap and that approximates the cost of our tagged amplicon sequencing assay, which is about 20 quid. And the advance from that has been to go back to P53. So this panel here just shows tagged amplicon sequencing for P53. This is a relative copy number assay. And what we're doing really is saying, if we can measure the allele fraction in a tumour sample, which of course we can, that's a synthesis between two things: the purity of the sample, how many tumour cells are there in it, and what's happening at copy number state at P53. But we can use those as a prior probability in a model that then attempts to fit absolute copy number across the entire genome. When you do that, you go from a default state of 0, 1, in other words, no copy of wild-type p53, one copy of mutant p53, 0, 1, all the way up to 0, 10, and you use an expectation algorithm to find the best fit across the entire genome. And that turns this sort of information into this sort of information. And it can be remarkable. I don't have this isn't the example I normally use, but so for instance, you may see chromosomes 1 and 2, not this case, but chromosomes 1 and 2 looking like they're diploid, but actually when you go to absolute copy, they're threefold, and the 2-copy gain here can be tenfold after you've done those fits. And so when we apply those types of assays to the BritRock cohort, so there's 130 patients here, both the original archival samples and the relapsed samples, we start to see something we've never seen before in high-grade serous ovarian cancer, which is some idea of structure around copy number. So here you've got this large group here with a near diploid genome, shown in blue. We've got higher copy here, up to 4 copy and higher. And then we've got a mixture of probably 2 or 3 groups here representing different states. And when we look at the patterns of that, we're starting to call these genome types. I hope that is not hopelessly optimistic. But what we're starting to see is individual patterns coming through, which I've shown here. So about 36% of the cases in the... Oh, and I should also emphasize that in the BritRock cohort, there's very close correlation between your baseline diagnostic genome type and your relapsed genome type, although we'll talk about heterogeneity in a second. So what we're starting to see is a predominant simple genome pattern, about 36% of the patients out of that cohort with large, either single arm or whole chromosome changes, but you can see cyclin E amplification very easily here on chromosome 19. We see a stepped genome that is more compatible with a breakage fusion bridge type pattern, more aberrations. We see tandem duplicator, which is a phenomenon where you do end-to-end reduplication of segments. This is about 4% of patients. And then this phenotype here, which was originally described by Joe Gray in breast cancer, which is called amplifier, where 5 or more amplifications at greater than 5 copy. And in this case, you've got over 10 amplifications, each well over 10 copy, on the background of quite an extraordinary amount of disarray. So this is just a breakdown at the moment. I haven't shown you the chromothripsis or the shattered genomes, which are just off the scale in terms of their aberrations. And this is the correlation with BRCA1 and BRCA2 germinalized status in these patients. Interestingly, we don't see any overlap between BRCA1 and BRCA2 in the tandem duplicator set, which definitely seems to be a feature of triple-negative breast cancer. And lastly, what is really great about this is that we can also do this on blood. So in the same way that I described a few moments ago, this is a patient from a biopsy from one of the BritRock patients. Their allele fraction was 32%. We get a very nice absolute copy number profile. Their plasma ctDNA allele fraction was 43%, and we also get a very nice absolute copy number fit. So this is work that is very recent and ongoing, but I think it is going to be important. It's important because the first time we have some way of actually looking at the mutator phenotype output in high-grade serous ovarian cancer. We think this could be thought of as a grammar for trying to understand what's happening. And of course, most importantly, it's immediately applicable to the large study populations and clinical trial populations we've got to tie up with other endpoints, particularly, for instance, response to platinum or PARP inhibitors. And I haven't got time to talk to you today, but there is already emerging evidence that different copy number profiles, either from SNP6 or foundation sequencing, can be used to predict response to rucaparib or other PARP inhibitors in the clinic. I think using whole genome sequencing, shallow whole genome sequencing, will give us a bit more power to dissect some of those mutator phenotypes. And clearly, gene expression signatures are going to be highly polarized by copy numbers. So that amplifier phenotype has hitherto not been analyzed in terms of what may be key drivers or key targets within those patients. Last couple of slides. I think there's one other thing that we have to do now in terms of management of patients with upfront disease and relapse, and that's really use Ascites as another liquid biopsy. We have done a lot of work around, for both gel and for BritRock, in biopsying patients at relapse, but biopsy is by no means straightforward, but it's often easy to get to Ascites, and from Ascites we get some really important things. So we get high cellularity tumour spheroids that have organizational structure, and we can use those for drug screens and functional studies. We can get single tumour cells, but we can also get fluid. And I told you a few moments ago that the fluid from Ascites is a very high and rich source of circulating DNA, mutant DNA, cell-free DNA. But we can also then profile Ascites spheroids, and I just wanted to show one slide about evolutionary change from absolute copy number. So this is the patient. He had a biopsy in January 2012. These are Ascites spheroids taken over time, and what these rectangles here are showing progressive loss around 3Q as the left-hand box, and 15p here. Sorry, 3p and 15p. Progressive loss over time. So, I think we're still well behind the curve for high-grade serous ovarian cancer compared to colorectal cancer, esophageal cancer, endometrial cancer, but I think we are getting there. Our approach is that we need to bring cheap, reliable biomarkers into the clinic, do it at scale because the discovery pieces still have to do. P53 IHC, critical. Circulating tumour DNA, I think, is a promising tool, and we now see that role being used for monitoring and minimal residual disease. And ultimately, we have to start getting measurement of important markers like cyclin E into the clinic and starting to do the adaptive trials of these markers. So with that, I'd just like to acknowledge the collaborators and the people who did the work. Most of the work I've shown you today is the work of Teddy, Jeff, and Anya Pishkors. But the circulating tumour DNA started on a very productive collaboration with my friend Nitzan Rosenfeld. Ian McNeish leads the Britrock study, and Martin Coble, very important in terms of the characterization of IHC. Thank you very much indeed.